So very quickly, I will be going over some steps in terms of the DNA replication process, and I'll be doing so with a little diagram to help me, along with the mini Kendrick Lamar to point out the things I discuss. So we're gonna kick things off with the topoisomerase, which essentially all it does is relieve the tension and strain that builds up ahead of the replication fork. As DNA is unwinding, its job is basically to make like cuts, preventing the DNA from being tangled up. So after that, we're we're at the replication fork, where the helicase unwinds the double helix of a DNA, and it does so by simply breaking the hydrogen bonds between the complementary base pairs, separating the two DNA strands into two single strands. So then the primase synthesizes short RNA primers found on the single stranded DNA template, making a starting point for the DNA polymerase to start at. And then the DNA polymerase reads the template strand from 3' to 5' in that direction, where it then synthesizes a new strand from the 5' to 3' direction. So when the DNA polymerase synthesizes the small fragments of DNA, the ligase joins them together its job essentially is just to seal the gaps between the Okazaki fragments, which is just like, again, the small sections of DNA.
Inside the double helix structure, there is two directions, 5', which is upstream, and 3', which is downstream. Nucleic acids can only be synthesized in the 5' to 3' direction, as the polymerase that assemble various types of new strands generally rely on the energy produced by breaking nucleoside triphosphate bonds between the 5' and 3' end.
The difference between a lagging and a leading strand is the lagging strand replicates discontinuously, forming short fragments, while the leading strand replicates continuously. Okazaki fragments are small sections of DNA that are formed during discontinuous synthesis of the lagging strand during DNA replication. Semi-conservative is when a double-stranded molecule of nucleic acid separates into two single strands and serves as a template for the formation of a complementary strand that, together with the template, forms a complete molecule.